All right, so this week we are talking about biological evidence, DNA, and also we will cover blood spatter in a separate recording. All right, so biological evidence is a good hazard for us as evidence technicians and CSIs. So do be cognizant of your PPE, your personal protective equipment. You're going to come into contact with a number of bodily fluids, blood, saliva, any secretions from any part of our body, and all can carry bacteria, viruses, anything that can make you sick. So do be very cognizant of your PPE and not just rubber gloves. All right, so blood evidence. Big question is this blood or not, and is it human blood? We have what are called presumptive tests that we will use, and it's not just blood evidence. I probably should have relabeled this slide now that I'm thinking about it. It should be biological evidence. Is it actually blood? Is it something else? Is it saliva, pee, semen, any colored fluid or colored stain that you find at a crime scene, depending on what you're investigating? Is it actually human in nature, or is it something else in nature? Is it biological at all, or is it something completely different? Is it a cleaning fluid, not even, some sort of human created stain? It's a stain from dinner preparation, whatever the case may be. So we have some different presumptive tests out there, and using these presumptive tests, what we can end up doing is seeing if we get a reaction to the stain, and does it test positive for being blood or not? Does it test positive for being semen? Does it test positive for being another human biologic or not? And then also, is it human or is it an animal source, depending on the test kit you get. Most all of these test kits these days, they essentially, pardon my description here, but they look like a home pregnancy test. You take a Q-tip, you touch it into the stain, you touch it onto the piece of cardboard, you wait a couple minutes. If it's got a plus sign, yep, we've got blood or semen or urine or saliva or whatever biologic the test kit is. If it's got a negative sign, it ain't what we're looking for. It's something else. It's ketchup. It's cleaning detergent. It's something that is not what we are looking for that's biologic. All right, so the big thing with biologics, a lot of people are concerned with, is DNA. DNA means deoxyribonucleic acid. I always have to look at the spelling to remember it. Deoxyribonucleic acid. We've known about DNA for a lot of years, but it wasn't until 1984 where we actually had somebody see it, literally actually say, this is DNA. We knew there was some building blocks down inside of our cells. There was something else in there, down in our chromosomes, but Alec Jeffreys in '84 was the actual first person to really truly see the DNA and able to identify it. And with DNA, we're able to make a lot of our biological evidence become individualistic, because everybody has different DNA, except for identical twins. Identical twins were one egg that split into two, and then instead of sticking together and growing into one human being, for whatever reason, they drift apart, and we get two human beings. So they have identical DNA because they came from the same sperm and the same egg. Non-identical twins, two eggs, two sperms, two people growing in the womb. Now, identical twins, as we mentioned before, do not have the same fingerprints because fingerprints are not encoded in your DNA. Fingerprints come from your interaction with the environment in utero. Now, using the variable number tandem repeats, we can actually start looking at some different things to see whether or not this DNA is one person or somebody else. Now, the interesting thing about DNA, the vast majority of our DNA code makes us human. Gives you two eyes, gives you ears, puts the nose here, puts the mouth here, puts the teeth in the mouth, puts the hair up here, etc., etc., etc. That is what most of our DNA is. Now, the unique things in our DNA, the things that cause me to have my blue eyes, and the things to say that my nose is going to have this shape, and it's going to, my ears are going to be these big floppy things that I have. And the fact that my hair is brown and that my hair has a little bit of a curl to it, so I like to keep it short. That is the stuff that is unique to me. That is the fact that my, mom and dad, part of their genetic code was imprinted onto me. Most of their genetic code is the fact that you have the two eyes, you got the nose, you got the mouth. But it was the fact that we had genetic markers for blue eyes and this shape of nose and my mouth to be this big and my teeth to be what they were or what they are. That is almost 1/10 of 1% of us. 99% of our DNA, all the same. It's just a small little segment. Gives you the blue eyes, gives you the nose, gives you the teeth, gives you even my pasty skin that I go out in the sun and I'm usually pale as can be and I'm either pale or sunburned. That's in my DNA. That makes me unique from you. So that's what we're looking at in DNA typing, is just this little tiny percentage, very, very small percentage. But it's funny, DNA is very hardy. Even improperly stored, we are still getting DNA hits on cases, but improperly stored DNA can degrade. So we want it properly stored. And we're solving cases from before the time we knew that there was DNA, where we're making identifications. As a matter of fact, a couple of scientists are looking at the serial killer, Jack the Ripper, 100 years ago. And there was some DNA found at some scenes, and they're thinking that they might have a possible identification. A couple of people, some family traits, and they've really narrowed down who they think Jack the Ripper might have been. Kind of a cool thing when you think about it. But we can use DNA to identify human remains. So we have folks who think that their loved ones went missing in Colorado, and a skeleton has been found in Colorado. And now we can actually run DNA typing and see if that is that person, because again, their DNA is half mom, half dad. And so we can run a swabbing of the inside of mom or dad's mouth, and then we can check and see, is this person's DNA profile, is half of it line up with mom's profile? If we get both mom and dad's profile, then we can say, hey, does half their profile line up with mom and half their profile line up with dad? And look at that, 2 halves is a whole. All right. A little out of order here, sorry. So our presumptive testing, not only can we do presumptive testing, but also we can use some of our presumptive testing chemicals. Instead of using like the home pregnancy test card device, we have some chemicals that we can put out at our crime scenes and find hidden biological evidence. So here we've got a carpet that has been cleaned up, and even the wall, and taking and treating this with luminol, which is one popular chemical. There's another chemical, Blue Star, that has become very popular. It's not on the list here, but what we can end up doing is treat surfaces that it's hard to see blood, or blood has been cleaned up on, but not cleaned up good enough. And we end up having, in a darkened condition, biologics glow. And then we can find where our hidden biologics are. So how do we go about collecting biological stains? Q-tips. If the stain is wet, you just put the Q-tip into the stain. If the stain has dried, then we take little pipettes of water with us to our crime scenes. We prefer to bring our own water. It's not necessary, but I think I've said it before here in these videos. I'm a little anal retentive when it comes to my crime scene, so I like to bring my water in so that I know it's biologically neutral. There's no additional DNA in there. We put the water on the swab, then we rub the swab into the stain, and then the stain is collected. Allow these swabs to air dry as best you can before sealing them. Sometimes we will prepackage them at the scene, transport them back to the PD, and then air dry them for final packaging. So allow air dry to package, collect comparison samples on scene. So if you've got a victim of a sexual assault or a victim of a battery, you need to collect a sample of their DNA at the scene. If you have the victim of a homicide and they've got living family members living with them, you need living family members' DNA also collected so that we can say whose DNA belongs at the scene versus our suspect's DNA that does not belong at the scene. And then also we need, if we have a living victim in assault or a battery, we need to collect their DNA so that we can say, okay, this blood puddle is my victim's DNA, this blood puddle is my suspect's DNA, this is the blood puddle, the suspect DNA puddle is the one that we're going to send to the lab for testing. All right, so that is the basis here of biological evidence and the DNA structure of it. The next recording after this one, we will be talking about blood spatter, which is kind of an interesting and cool topic that some folks like. All right, see you in a bit.